Hi everyone, in my last video we talk about the lipid part of the plasma membrane. If you have not seen it yet, you can find the link in the description box and watch it. Now in this video we will be discussing the protein part of the plasma membrane. As you already know, the proteins are the other major constituents of cell membrane, constituting 25 to 75% of the mass of the various membrane of the cells. So the basic structure of membrane proposed by Singer and Nicholson in 1972. This is the fluid mosaic in which proteins are inserted into the lipid bilayer. So the depending on the cell and particular organelles within the cell, a membrane may contain hundreds of different proteins. Okay. So each membrane protein has defined orientation related to the cytoplasm, so that the properties of one surface, okay, so one surface of the membrane are different from the other surface, and this type of symmetry is called as sideness. Okay, so you can see in this picture, this is the fluid mosaic model membrane structure. In this plasma membrane, for instance, those parts of membrane proteins, okay, and those parts of membrane proteins which interact with the other cell or with extracellular substances are exposed to the extracellular space, whereas those parts of membrane proteins that interact with the cytoplasmic molecules are exposed to the cytosol. So based on membrane proteins can be grouped into three distinct class. Okay, it depends on familiarity with the or relation to the lipid bilayer. So first is the integral protein or transmembrane or intrinsic. So integral proteins are transmembrane proteins, that is they pass entirely through the lipid bilayer and thus have domains that protrude from both the extracellular and intracellular side of the membrane. And most of the integral membrane proteins are globular proteins. Some integral proteins have only one membrane spanning segment, whereas the other has multi-spanning. For example, glycophorin A, RBC, red blood cell, and second is band 3. Okay. Like the phospholipid, as we learned in my previous video, lipid. Okay, so phospholipids of the bilayer integral membrane proteins are also amphipathic, having both hydrophilic and hydrophobic portion. So those portion integral membrane proteins that reside within the lipid bilayer, the transmembrane domains, tend to have hydrophobic character. Okay. So amino acid residues in transmembrane domain form van der Waals bonds with the fatty acyl chain of the bilayer. So that's why we see which seals the protein into the lipid wall of the membrane. So these non-embedded domains tend to have hydrophilic, this one, a surface that interact with the water soluble substance like hormones, okay, low molecular weight substrate and other proteins. So this is how the transmembrane proteins form. Okay, so the membrane spanning portion of transmembrane protein are usually helix of 20 to 25 hydrophobic amino acids that are inserted into the membrane of endoplasmic reticulum during the synthesis of the polypeptide chain. That means that when the ribosome synthesize the polypeptide chain, they insert it into the endoplasmic reticulum's lumen. Then reticulum's vesicle transfer to the endoplasmic reticulum's Golgi apparatus compartment, and they transport through the vesicle into the Golgi apparatus, and some slight modification it has happened. Okay, like glycolipid and glycoprotein, and they transfer the Golgi apparatus vesicles to protein into the plasma membrane. So now, so the integral membrane proteins of red blood cell. So the two major integral membrane proteins of red blood cell, first one is glycophorin A and second one is band 3. So this is the transmembrane protein structure both. Okay, so glycophorin is a small glycoprotein that contains the 131 amino acid, which is molecular weights about the 30,000. Okay, and their single membrane spanning alpha helix of 23 amino acid, and it's heavily glycosylated. Okay, here N terminal site about 50% carbohydrates. Okay. The other major transmembrane proteins, that is the band 3. The main function is the Ni transporter responsible for the passage of bicarbonate and chloride ions across the red blood cell. So the band 3 polypeptide chain has 929 amino acid with 14 membrane spanning alpha helic region. This is a multi spanning region. With the membrane diameters of band 3 form globular structure containing internal channel through which ions are able to travel across the lipid bilayer. So now second is the peripheral proteins. So peripheral proteins that are located entirely outside of the lipid bilayer on either cytoplasmic side or extracellular side. So which are associated with the membrane by weak electrostatic or non-covalent bond. So this reason they can easily extracted from the cell membrane by the applying the salt solution, high concentration of salt solution. Okay, in actual fact, the distinction between the integral and peripheral proteins is blurred because many integral membrane proteins consist of several polypeptide chain. Okay, some that penetrate the lipid bilayer and other that remains on the periphery. Okay, so this is the image of plasma membrane of human erythrocyte. So model of erythrocyte plasma membrane is viewed from the internal. This is the internal. This is the internal side. This area is internal surface, showing the integral protein embedded in the lipid bilayer and the arrangement of peripheral proteins that makes up the membrane's integral skeleton. This looks like skeleton. The band 3 dimer, this one shown here is simplified, and band 4.1. Band 4.1, which is protein stabilized actin spectrin complexes. And also these proteins provide mechanical support for the membrane and function, okay, as anchor of integral proteins. Some of the other types of peripheral proteins on the internal plasma membrane surface its function is enzymes, specialized coat and factor. They transmit transmembrane signals. Okay. And so in this ankyrin and protein 4.1, both are the peripheral proteins. Okay, so ankyrin serve is the principal link between the plasma membrane protein. Okay, so plasma membrane proteins, that is a band 3. Okay, band 3 is the integral plasma membrane protein and binding to the spectrin. Okay, spectrin to the integral membrane protein band 3. So now in a okay, so moreover in protein 4.1, the link between the membrane and the cytoskeleton is provided by the band 4.1, which binds to the junction of spectrin. This is the spectrin. This is the spectrin, for example. Okay, spectrin, actin, as well as to the glycophorin A. Okay, so glycophorin. This is the main function of ankyrin and protein 4.1. Okay, so third one is the lipid anchor protein. So lipid anchor protein that are located outside the lipid bilayer, okay, on either the extracellular or cytoplasmic surface, but are covalently linked to the lipid molecules that is situated within the bilayer. Okay, several types of lipid anchor membrane proteins can be distinguished. So numerous proteins present on the external face of the plasma membrane are bound to the membrane by a small complex oligosaccharides. okay, linked to a molecule of phosphatidyl inositol that is embedded in the outer leaflet of the lipid bilayer. So this is the phosphatidyl inositol, and they phosphate head binds to the oligosaccharide. Oligosaccharides binds with the protein. Okay, they are discovered when it was shown that certain membrane proteins could be released by the phospholipase. Okay. So another group of protein present on the cytoplasmic side of plasma membrane is anchored to the membrane by one or more long hydrocarbon chain. So normal cell portion of protein PRPC, okay, is GPI linked molecules are various receptors, enzyme and cell adhesion proteins. Now the rare type of anemia, okay, the disease result from deficiency in GPI synthesis. Okay, that makes the red blood cell susceptible to lysis. Okay, so another type of lipid anchor protein, that is the 2 protein associated with the plasma membrane in this way, SRC and RAS. So this is the 2 proteins have implicated in the transformation of normal cell to malignant state. So this is all about the today video. Okay, I hope this will help you. Thank you.